Welcome students. Today we are going to discuss about the different fermentation types. In this episode we are going to cover the topic on batch, fed batch and continuous fermentation. If we have a closer look at what these three terms mean, we will see that a batch culture or a batch fermentation involves the use of the components that is required for fermentation at once. That is, we fill the entire bioreactor only once, inoculate it only once, and then wait for the products to be produced, then can take it out. While in case of a fed batch fermenter, what is done is, it is pretty similar to the batch fermentation. Everything is incorporated only once, but provisions are kept so that some of the substrate can be added at times later or even throughout intermittently. While in a continuous fermentation, what is done is substrate is continuously added and product along with some substrate and cells are continuously derived of. Now in this picture, the X stands for biomass, which is the cells. S stands for substrate, that is the component which is to be converted to product by the cell. P is obviously the product, which is the outcome that we are supposed to get, and T refers to time. Now if we look into this batch culture, we will see that everything considering biomass, substrate as well as product are time dependent. While in fed batch also, because it is very much similar to batch, all the three parameters are again time dependent. While in case of the continuous culture, it is different and there is no time dependence. That means at any point of time, you take any sample from here, they are not time dependent. They will be the same, and that is why we call it a steady state fermentation. We are going to have a broader discussion about these three types in the next few slides. Let us first begin with the batch fermentation. It is considered as a closed system because just after the first incorporation, there is no allowance for input of any substrate whatsoever during the entire course of fermentation. For batch fermentation, it is essential that we realize the growth curve of a typical bacteria. Now, here is represented the typical growth curve that a bacteria follows during the entire course of its life cycle. Now, in a batch culture, which is a closed culture, this whole of the growth curve can be observed. First, we begin with the lag phase, then there is a log phase called as exponential phase, then the following phase is called as the stationary phase, finally the death phase. Now interestingly, in fermentation technology and in industries, we are not much bothered with the death phase. We begin with the lag phase here. The lag phase, although shows no importance to us as such, but is highly important in case of any culture, be it industry, be it the lab. Let us see what happens. If we consider this as a single cell, which is in the lag phase, and this is the surrounding medium, these are representing the nutrients which must enter the cell so that the cell can utilize it for its growth, division and product formation. Now, there is some point of time which is required for the cell to actually be ready or prepare itself so that these molecules can enter the cell, and this is the lag time of a culture. Why is the lag time important in industry? We must realize that if the lag time is more, then we are simply wasting time, and there should be something done so that the lag time can be reduced to as less as possible. So industrial microbiologists or biotechnologists, what they do is they generally use the inoculum from a log phase culture of a seeding tank, meaning the earlier culture that they take is from a log phase culture, and maintaining the medium composition approximately same for growth medium and production medium will generate a very low or very less lag time. Moving into the next phase of the growth curve, that is the log phase. So in this case, the cells which were in the lag earlier have adapted and are taking up nutrients and now are ready to go into the log phase, which we generally know by the name of exponential phase, because this is where the cells are actively dividing exponentially. Let us look a closer picture here. What is happening to the cell? We can see the inner diameter here, which is the usual size of the cell when it is in the lag phase. Now, because it has taken enough nutrients into the cell, it has grown in size. The growth of a bacterial cell obviously is kind of negligible if you compare with the growth of other higher cells organisms. But correlation of bacterial growth is represented by the division rate, which definitely will follow soon after the bacteria attains a specific size enlargement as well. This happens and then the bacteria starts dividing, first multiplying its DNA and then itself dividing into two. This phase is the log phase that we are discussing, and here we can actually see what is exponential growth. One cell divides into two, and in the same amount of time the two cells will divide into four, likewise 4 to 8 and finally 16. So as we can see here, the cells are progressing in numbers geometrically. So the increase is pretty high, and that is why a good kind of a slope is produced. Now, log phase is very, very important for a biotechnologist or an industrial microbiologist. What we can do here is study the growth kinetics of the organism that we have chosen to be used for the product formation. Until and unless we understand the kinetics, we will not know when exactly to begin and when to end with the product. Thus, the calculations so far that we know of for growth calculation and kinetics is expressed and understood all from the log phase of a batch culture. We cannot do that easily in a fed batch or a continuous culture. This is the best place where we can do it. What exactly we are calculating? Now, rate of increase in cell biomass and rate of increase in cell number is what we can correlate with. Now, there are two different parameters here. One is a biomass, the other is a number. We will use biomass for any mycelial natured cells like fungal cells or actinomycetes, where we cannot correlate with numbers. For others like typical bacteria and other cases of yeast cells where we can correlate with numbers, we can do with the cell number. So whatever it is, actually we are concentrating on the increase in the cell, whether it is by weight or by number. And the rate is significant here. At what rate are they growing? So that will be given by dx by dt, which will be the factors, that is mu into x if it is biomass, and dn by dt is equal to mu into n if it is cell number. Now, what exactly is this mu, which is a common factor that we can see here? Mu is a specific growth rate. X stands for the biomass, n stands for the cell number. Mu is the specific growth rate, and it is the rate of growth where a specific substrate is implied. It is not the best growth. Now, we need to understand mu because in industry, media is not the best for its growth. It is meant for actually production, and that is why specific mu or specific growth rate will be different from maximum growth rate, that is mu max, as we can see here. Mu max is the best growth rate. So Monod actually defined the equation, and that is stated as the specific growth rate can be easily calculated if we know the maximum growth rate, and the equation says that mu is equal to mu max into S divided by Ks plus S. What is this S here? This is the substrate concentration, and Ks is the substrate specific constant. Now, if we know mu, we can substitute this mu and multiply by x, so that we get dx by dt is equal to mu max into S into x divided by Ks plus S. And this dx by dt is nothing but this mu into x, which will tell us what is the rate of increase in cell biomass. Why do we need all this? Until and unless we know the cells are growing and we cannot calculate, we won't know whether we will get a product and if the product that we get will have good amount of yield. So this is very, very essential in any sphere, and whether we carry out a continuous culture or a batch or a fed batch, we need to know the growth kinetics, which will be obtained only from batch culture and from the log phase. Moving into the next phase, stationary phase of the growth curve of bacteria. This is the stationary phase. It parallels the time axis and looking like it may seem as if there is no growth. But friends, let us not forget that in the stationary phase also growth is there, which is equaled by the death also. That means some cells are dying, some cells are growing, and there is a balance which shows that actually there is no growth, but growth remains. And this phase is really important if we consider the case of some secondary metabolite production in case of the industries. In the industries, we will see the production of secondary metabolite, antibiotic being a very common example. But first let us see what is happening to the cell. A cell which is in the secondary phase of growth is almost grown completely, filled entire of its life cycle, and nutrients, although are available, they are not feeding and they are not taking in. So they are almost kind of not growing at a very fast rate. They are existing, and as time passes by and the surrounding becomes little more toxic to them because many cells are dying and their products are being released, these cells will eventually also die and move into the death phase. It seems to be very unimportant, this phase, but we are looking forward to this phase because the important antibiotics, many kinds of flavonoids, others plant products are obtained from this secondary phase. This secondary metabolites may include pigments like carotenoids, anthocyanins. It may include alkaloids like morphine, then terpenoids, various types, some essential oils like lemongrass oil, some toxins, lectins, drugs, and certain polymeric substances. Now, basically, if we also look into the closer look of these secondary metabolites, what are exactly these secondary metabolites? They are different from primary metabolites in the sense the primary metabolites are required for the life and existence of a cell. They will always be produced if the cells are growing. Some examples would be citric acid, lactic acid, which they produce for their growth. But there are other substances which a cell normally do not produce but has the ability to produce and produces if required. That is, if the environment calls for it. And these are the secondary metabolites. Some plants may also produce the secondary metabolites which are chosen, and accordingly industries produce huge amount of these secondary metabolites. And we target the stationary phase for the production of these secondary metabolites. Finally, we come to the death phase, and it is to note that the death phase has no role in industrial sphere because once the organism dies, they are of no importance, and so we basically do not do anything with the death phase. So this is where we have a good picture of what a growth curve importantly looks forward to in an industrial sphere. But let us now have some idea about the formation of product and the growth phases, or rather growth associations. There are three curves here which we can see. Now X like the biomass and P is the product. What happens if the product that we are deriving is growth associated, means they are primary metabolites, then they almost parallel the two curves are almost parallel, because as the cell number increases, the product formation increases. As they decline, the product formation declines because they are primary metabolites. Sometimes we cannot explain, and there is a second category, and they are not much dependent on the growth, but they are slightly dependent. So we can see the second curve here, that the organism is having a typical lag, log, and a stationary, while product formation does not necessarily correlate or go into parallel with the growth curve, but there is an increase of the product formation as the cells increase in number or moves towards the end of the phase. While in case of non-growth associated products, or as we have just explained, the secondary metabolites, see the cells increase in number. They are almost towards the stationary phase when the product formation starts. So this is the secondary metabolite production, which occasionally happens in mostly the stationary phase. Now we are looking into some pictures of the batch fermenters which are commonly used in the industries. To begin with, we start off with a agar slope, which is an example of a batch fermenter. Why we say here fermenter? Because the organism is growing, media is there, and that is why this can be a simply smallest size reactor or a bioreactor where the organism is maintained in the form of agar slopes. Then we culture in a shake flask, most often in the lab. It can be a small bioreactor as well, but commonly shake flasks are taken, and these are also batch fermenters because they are inoculated once and they are left for the product formation. Then the downstreaming can be done when the product is produced. No incorporation of media takes place in between. There is a seeding tank. It is often used to increase the inoculum amount which can be inoculated into a large plant scale bioreactor. So this is also example of a batch bioreactor. And finally, we do have plant scale bioreactors which operate in the batch mode, and you will be surprised to know that the maximum type of reactors which are available are batch type reactors. Although we have advantages of fed batch and continuous fermenters over batch fermentation. The advantages will tell us why we are so much using batch fermenters. The initial capital expenditure is less. This is very important. An industry will flourish only if the initial capital cost is less, so that more profit can be procured. The second advantage is, by chance there is a contamination, then the whole batch can be simply terminated without much loss and a fresh start can be made. So these two advantages have made the batch fermentation so very more important and widely used. But there are certain disadvantages of the batch fermentations as well. Let us note some of these. Now basically, we cannot obtain a steady state, means the cell growth will not be constant. There will be a time when the cells will be kind of deprived of nutrition because they will be exhausted. We are not introducing fresh media and they will die off. So we cannot obtain a steady state. Then the operation of a typical batch fermenter requires several stages, like charging of the fermenter, means introduction of media, inoculating the media, sterilization of the fermenter as well as the media. Then it will require some time like product formation, harvesting or recovery, and cleaning of the vessels. During all this time, when we carry out certain of these operations, the bioreactor probably is not in function all the time, and that time is considered as the downtime of the fermenter, means the bioreactor is not operative, and as such there is some loss that can be procured. This can be explained well if we are comparing a fed batch or a continuous bioreactor with a batch bioreactor, because they are more advantageous because they are constantly performing. Then the other disadvantages of the batch reactor also includes batch to batch variability. One batch may have a different product, the other batch may slightly vary, and that gives rise to variability. And running costs are high because more personnel are involved in the operation of a batch fermentation. Now, we have learnt about batch fermentation, but what are the products that we can derive from a batch fermentation? And those are the alcoholic beverages, the ethanol that we get, and also the alcoholic beverages that we get comes by batch fermentation because they are the actually primary metabolites. Amino acids, enzymes, and organic acids, all being primary metabolites, will obviously come from batch fermentation very easily. Next, we are moving on to the second category, that is of fed batch fermentation. This is not exactly a closed reactor. It is considered as a semi-closed reactor because, like batch, the inoculum as well as media are added at once. But at times, in between or intermittently, some or one of the substrates can be added. Nothing is taken out, and as such it is not an open system. As we can see here in the diagram, that fresh medium, there is the port of fresh medium which is allowed that during the course of fermentation, if required and when required, fresh medium may be added. There are two types of fed batch bioreactors. The first is a fixed volume fed batch. The second is a variable volume fed batch. Why do these volume changes occur anyway in fed batch? Obviously, we have to understand that if we are adding on some amount of nutrients and not taking out anything, then the reactor volume will increase. And this is a major problem because the bioreactor cannot be completely filled and we are also introducing some more of medium. So the volume may increase and cause problems like pressure buildup and other viscosity problems. To eliminate those, we generally try and see that we can go for fixed volume fed batches so that the substrate can be incorporated but the volume does not increase much. And this is obtainable in certain instances. Like for example, here is a typical photobioreactor, and this means if light is provided, then the cells will grow. So light is a limiting factor. If you are not allowing light, then the photosynthetic organisms will not grow. They will not photosynthesize. And that is the reason a photoreactor is a very good example of a fed batch, because you are controlling the growth of the cells within the bioreactor by supplying or not supplying a particular substrate. This is light here in case of photobioreactors. There are others like wherever there is a problem of oxygen. If oxygen is used as a substrate or any other gas which is used as a substrate, if that is introduced, even then the bioreactor volume does not change, but you are incorporating extra material, extra substrate. So this becomes a typical fixed volume fed batch. The second is variable volume fed batch, which is the common types where the volume will increase, and the disadvantages of the volume increasing can be solved by adding discrete or concentrated forms of the substrate that should be added. Now we are seeing some of the advantages of a fed batch fermentation. Most importantly, wherever there is catabolite depression and viscosity or toxicity as problems, fed batch is used. And typical examples where fed batch is in use introduces the production of Baker's yeast and penicillin, the antibiotic for use, and hepatitis B vaccines. There are so many more, but these are the major ones where fed batch mode of fermentation is used to bring us the product. Then we are moving on to the continuous fermentations. These are obviously the best as can be understood, but the problem is the initial expenditure is very, very high. The mode of operation is simple once the process has begun. What happens here is constantly fresh medium is added and there is constant removal of a washout, and as such the steady state within bioreactor is maintained. The cells are growing in the same phase and the substrate concentration and other concentrations are almost the same throughout. There are some types of continuous culture possibilities, or rather how we can maintain. First is a chemostat, which is regulated by controlling the concentration of the limiting nutrient. The second is a turbidostat, which means that regulation is brought about by the use of optical density measurements where biomass can be regulated. And thirdly, there is a biostat, where the systems are there for monitoring biomass other than optical density, for example, carbon dioxide gas produced by the organism. So this is how we can achieve continuous fermentations, and that is important in a lot of alcohol production, ethanol production, and obviously the best mode that can be available. Where cost is not a factor, we can always go for continuous production, but if cost is a problem, then batch and fed batch is often preferred. Then we have seen these three processes, and we will just briefly define what are the single phase and the dual phase fermentations. Most often in a bioreaction, there are at times the requirement of one or a single fermenter. That means sometimes only primary metabolism is important, where we use a bioreactor which is single in phase, while at times two are kept together, say for example product from this will be separately used for conversion in another bioreactor. There we are commonly using a dual phase bioreactor. So this gives us a complete picture of how or what are the different types of bioreactor and processes of fermentation from where several products can be procured. This is interesting to note that it depends on the product which type we are going to choose.